Your DNA might be fixed from birth, but how it's read is anything but static. That's where epigenetics comes in, the study of how your genes can be turned on, off, or dialed up and down like a molecular mixing board. But what's truly remarkable is that these switches aren't flipped in isolation inside the cell's nucleus. Many are influenced by what's circulating in your bloodstream. In other words, the messages your DNA gets aren't just written in your genome, they're delivered by molecules traveling through your blood every second of the day. Blood, as it turns out, is more than a transporter of nutrients and waste. It's a biochemical telegram service for your entire body, and some of its most potent messages are epigenetic. Hormones, microbial metabolites, inflammatory cytokines, oxidative stressors, and nutrients all affect the way your DNA is tagged and interpreted over time. These tags include DNA methylation, where methyl groups attach to cytosine bases in the genome to suppress gene expression, and histone modifications, which alter how tightly DNA is wrapped around histone proteins, essentially loosening or locking up access to entire regions of your genetic code. Let's start with inflammation. When chronic low-grade inflammation, inflammaging, as it's called, floods the bloodstream with cytokines like TNF-A, IL-6, and CRP, it doesn't just damage tissue. It actually shifts the epigenetic architecture of your genome. These molecules can recruit enzymes like DNMTs, DNA methyltransferases, or DACs, histone deacetylases, to specific sites in the DNA, effectively reprogramming immune cells, turning on pro-inflammatory genes, and silencing genes involved in repair and regeneration. This creates a vicious cycle. Inflammation causes epigenetic changes that promote more inflammation, which in turn accelerates aging. Now enter the metabolite messengers. Acetoadenosylmethionine, SAM, a compound that circulates in blood and is derived from methionine and folate metabolism, is the primary donor of methyl groups for DNA methylation. If SAM levels are low, say, due to poor diet, oxidative stress, or mitochondrial dysfunction, methylation patterns can become erratic. This may lead to hypomethylation of oncogenes, bad news for cancer risk, or hypermethylation of tumor suppressor genes, also bad news. Your blood's metabolic state, in other words, is constantly tweaking the knobs on gene expression, sometimes with life-altering consequences. Then there's beta-hydroxybutyrate, BHB, a ketone body produced during fasting or low-carb diets. Far from being just a fuel source, BHB acts as an HDAC inhibitor, which means it can loosen up DNA by keeping histones acetylated. This promotes the expression of genes involved in stress resistance, longevity, and metabolic efficiency. It's one of the reasons fasting and caloric restriction have been linked to anti-aging benefits, because they don't just starve you, they epigenetically reprogram you through blood-borne messengers. Another class of molecules with surprising epigenetic influence are short-chain fatty acids, SCFAs, like butyrate and propionate, produced by gut bacteria and absorbed into the bloodstream. Butyrate, in particular, is also a potent HDAC inhibitor and has been shown to reduce inflammation, enhance neuroplasticity, and support gut barrier integrity by changing how genes are expressed in epithelial and immune cells. So, yes, your gut bacteria can flip your genetic switches from the safety of your bloodstream. More exotic still are exosomal microRNAs, tiny strands of non-coding RNA packaged in vesicles and released by cells into the blood. These microRNAs travel through circulation and can silence specific genes in distant tissues by binding to messenger RNA, mRNA, and preventing it from being translated. Think of them as stealthy genetic silencers dispatched from one part of the body to quietly influence another. Their patterns change with age, disease, stress, and diet, and many are now being studied as biomarkers or therapeutic targets for aging and cancer. Oxidative stress, too, leaves its fingerprint on your epigenome. Reactive oxygen species, ROS, in the blood can oxidize DNA bases, which not only leads to mutations, but can also alter methylation patterns. Some oxidized bases are more likely to lose methylation marks, altering gene expression in unpredictable ways. Worse, chronic oxidative stress can damage the enzymes responsible for maintaining proper methylation, throwing the whole system out of balance. It's like graffiti on your genome, with lasting consequences for how genes behave. And then there are the rejuvenating factors, made famous by studies on young blood transfusions in mice. Compounds like GDF11, growth differentiation factor 11, and oxytocin have shown the ability to restore youthful gene expression patterns in older animals by traveling through blood and signaling changes at the epigenetic level. While the hype around these molecules is far from settled, the concept remains powerful. Age is not just written in your DNA, it's written in how your blood tells your DNA to behave. All this points to a profound truth. Your blood is the narrator of your genetic story. The molecules it carries are constantly rewriting how your DNA is read, flipping switches that determine whether you heal or scar, thrive or decline, remember or forget. Epigenetics is the interface between your environment and your genome, and your bloodstream is the courier, delivering chemical instructions shaped by your food, your stress, your microbiome, your hormones, and your age. Which means aging itself may be as much about blood chemistry as it is about genetic fate. If we can identify and manipulate the right circulating factors, whether through diet, lifestyle, targeted drugs, or even engineered exosomes, we may be able to rewrite the script of aging at the epigenetic level, one methyl group at a time.